Okay, my computer is a little bit overworked at the moment. It's still trying to finish the lesson for the lecture. And so it'll be really slow for the next 5 or 10 minutes. Are there any questions? If there's no questions, let's begin. So today is August 4th, and we're supposed to be doing Lab 8, identification of gram-positive organisms. We do have some case studies in today's lab as well. That. I think I'm going to close the syllabus down as my computer is overworked as it is. Let's see if this is the right one. This is not the right one, but it'll do. Lab 8, identification of gram-positive organisms. We have some gram-positive organisms which cause human disease listed up here. There are other gram-positives, but these are the ones we're going to discuss today. Staphylococcus, Streptococcus, Enterococcus, a little bit of Clostridium, Bacillus. You know, I don't think we talk about Corynebacterium or Listeria. There are some video clips today, and we're going to be doing some case studies. To do the case studies, we have a gram-positive dichotomous key. The last two are provided as separate files in Canvas. Let's see. Are you seeing anything yet? There's a question for you.
No.
Ah, guys, I was talking like you guys could see it, so let me get this going. I need to show you Clark Canvas, Clark College website anyways. Okay, able to see my screen now.
Yes, we can.
And this is going to take a while because my computer is so overworked, but just give it a minute. It's trying to load. There it is, Lab 8, module, the worksheet. And then we have 8 different case studies to Lab 8. And we have a dichotomous key for solving the case studies. Only use this dichotomous key on case study number 8. Do not use it on your unknown. Do not use it on any other lab. Any questions about any of that? Excuse me, I'm about to sneeze here. Well, hopefully I won't sneeze. Let's see, leave that open, although I probably should shut it. Hopefully this will finish. No, we're only 65% done with the lecture. All right, the learning objectives for this lab. understand the principles and how to interpret the results for the CAMP test, the Bialis-Sculen Auger Swamp, the SF Broth test, and the Optogen sensitivity test. Be able to use a item is key to solve case studies involving gram-positive bacterial infections. Okay, when specimens are received in a microbiology lab for a routine bacterial culture, they need to be cultured on a culture media that will facilitate the growth of the common pathogens. And then they have to be placed in an environment to support their growth. Microbiologists can use different colony morphologies and the gram stain to provide important information in classifying bacterial isolates. However, that will not be enough. The colony morphology and gram stain cannot be the sole definitive use to identifying the bacteria. Further biochemical tests are required. In lab 7, we learned about the catalase, coagulase, and oxidase test. In this lab, we're going to talk about the CAMP test, the Bialis-Sculen hydrolysis test, the SF Broth test, and the Optogen sensitivity test. The CAMP test is named after the 3 microbiologists who discovered it or made it, and that's Christie, Atkins, and Munch Peterson. So they took the first letters of each name, and then for Munch Peterson, they took M and the P to get the CAMP test. The CAMP test is looking at the effect that some beta hemolytic organisms have when they're physically grown near Staphylococcus aureus. There's a synergistic phenomena that is easy to detect on blood agar. And let me just show you. Figure 8.1. And that is some bacteria, when you're growing Staphylococcus aureus on the street like that, and the center is where the bacteria is, Staphylococcus aureus engages in beta hemolysis, making the clear zone around the Staphylococcus aureus. When we grow some bacteria near Staphylococcus aureus, like Streptococcus pyogenes, we see little of any enhancement of hemolysis by the Streptococcus pyogenes, shown here. It looks similar in this region as opposed to the region away from Staphylococcus aureus. Other bacteria have a mild enhancement, like Listeria. It is showing more hemolytic activity near Staphylococcus aureus than it is away from Staphylococcus aureus. And then with Streptococcus agaltia, we have great enhancement of the hemolytic activity near the Staphylococcus aureus compared to further away from the Staphylococcus aureus. And I don't know why, but usually the enhanced activity of the beta hemolysis takes the shape of an arrow. Now that, of course, is the stick of the arrow, but then this would be the arrowhead. And for some reason, the enhanced hemolytic activity takes the shape of an arrow. Even here, if you look at it carefully, that kind of looks like a small arrow. So this is positive for the CAMP reaction, a slight positive for the CAMP reaction, and then negative in the CAMP reaction. And you can use this to test different bacteria. Like Listeria is different from Strep. I'm just going to go A right there. And it's different from Strep pyogenes right here. Any question about any of that? All right. The next test we're going to look at is the bile esculin hydrolysis test. Bile esculin is an ingredient added to this tube, and the esculin will be broken down by the organism to esculatin. Esculatin is a very dark brown color, so that if you see any black or very darkening of the agar, about 50% or more, we call it positive for the bile esculin test. And then if there's no change in the agar, it's negative for the bile esculin test. Any question about any of that? So this test can be used to differentiate between Enterococcus and Streptococcus. Streptococcus species are negative. They do not hemolyze esculin. Enterococcus is positive. It does hemolyze esculin and turn a very dark color in the agar. Any questions about bile esculin? All right. The next test we're going to talk about is the SF broth test. The SF broth test is named after Streptococcus faecalis, which is actually now a misnomer because they used to call the species Streptococcus faecalis, but with the, what do you call it, the DNA sequencing of the bacteria, they now realize it's not in the Streptococcus family, and it's actually in the Enterococcus genus. And so the new name is Enterococcus faecalis, but this test is still called Streptococcus faecalis. Sorry to confuse you there, but it's not terribly important. Anyways, the SF broth test, which is what I'm going to call it, contains sodium azide and the carbohydrate glucose, and then a pH indicator. And it is purple in color to begin with. And if the bacteria are negative, they cannot produce acid products when growing in the SF broth, then it will be purple in color or negative. On the other hand, if the bacteria can ferment the bacteria, glucose, excuse me, not ferment the bacteria, ferment the glucose, then they produce acids and it will turn yellow in color, and that is positive for the SF broth test. The Streptococcus species are negative, and Enterococcus faecalis is positive in the SF broth test. Any question about any of that? All right. Like I said, you do not need to know that the Streptococcus faecalis or SF test is a misnomer. It should be called the Enterococcus faecalis test, but we've kept the old name, and so I don't know. We'll just call it the SF broth test. Oh, good. Looks like the lecture just finished. My computer will be speeding up now. All right. The next test, and I think the last test I need to talk about, is the Optochin sensitivity test. Optochin is an antibiotic, and some bacteria are sensitive to it, where you can see a zone of inhibition of 14 millimeters or greater, as it's sensitive to Optochin. Another bacteria is not sensitive to Optochin, this antibiotic, in which case you will see either no zone of inhibition or a zone of inhibition less than 14 millimeters. Okay, so remember that if the zone of inhibition is less than 14 millimeters, we consider it resistant to Optochin. This test is frequently used to help identify Streptococcus pneumoniae. This bacteria is sensitive to optogen, and so we can see the zone of inhibition of 14 millimeters or greater. And other alpha Streptococcus is not sensitive to optogen, and we say it's optogen resistant. And let me state that all of these Streptococcus and Streptococcus pneumonia, when we grow them on blood plate, it is showing alpha hemolytic activity. Shown here, where you can see a darkening around the bacterial growth, but it is not a clearing. So this is alpha hemolysis, not beta hemolysis. Any question about that? All right. So when you're identifying the bacteria, you frequently will use a key to help identify the bacteria. And we have a dichotomous key to help you identify the bacteria for the case studies for this lab. So you can read about the dichotomous key. I don't know if it's this one or similar to this one, but we have the gram-positive stain or gram stain. And here we're only looking at the gram-positive cells, but that crosses out the gram-negative. And then the gram-positives, we then do the cellular morphology. There will be a gram-positive bacillus and a gram-positive cocci. And if our unknown is a gram-positive cocci, we cross out the bacillus. After having the gram-positive cocci, we then do the catalase test. And if they're catalase positive, we know that they are Staphylococcus, meaning in the Staphylococcus genus. If they are negative for the catalase test, we know they're either Streptococcus or Enterococcus. So if it's positive for the catalase test, we then do the coagulase test. And if it's positive for coagulase, it's Staphylococcus aureus. If it's negative for coagulase, it's some other staphylococcal species other than Staphylococcus aureus. And then if we do the novabiasin test, if it's positive for novabiasin, it's Staphylococcus epidermidis. If it's negative for novabiasin, then it's Staphylococcus saprophyticus. And that's how you use the dichotomous key. Any question about any of that? All right, so in the laboratory exercise, you're not physically going to be performing any of these activities. However, you will be held responsible for knowing how to perform and interpret them. We do have some video clips for you to watch. There's a video clip covering both the SF broth test and the bile esculin test. So watch those videos. And then we have a video covering the CAMP test. All right, let's go to the worksheet because we're actually now in the worksheet. So that's the first thing you should do for the worksheet, watch the videos. The second thing you should do is fill in this table here. Review how to interpret the following test by completing the chart. You will need to refer to some previous labs as well as this lab, like the Shaler-Fulton stain or an endospore, which was discussed in a previous lab. And I think that was actually 2 labs ago, not the last lab. For the gram stain, the results are filled in for you. For the gram stain, for cocci and bacilli, if you get a gram-positive result, the cells will look purple. And if you get a gram-negative result, the cells will look pink. We'll put in what a positive result for the Schaefer-Fulton stain for endospores will be, and then put in what the negative result is. Do the same for a capsule. What does the capsule look like for a positive result? And what does it look like if there is no capsule, a negative result? Put in for the catalase result, hydrogen peroxide. I think that was discussed in our last lab. And then negative result. And I'll tell you what we have for the negative result is no bubbles. Put in for the coagulase test, the oxidase test, the CAMP test, which is in today's lab. Positive is enhanced hemolytic activity. And then the negative test is no enhancement of hemolysis. Polyskeurin, put in the positive result and the negative. SF broths, positive and negative. And then the Optogen test. Be careful on this one. You can't say positive has a zone of inhibition and negative is not. You have to say the inhibition is below a certain size to be negative. All right, then view the video on how to develop and use the dichotomous key. You're not going to be using this dichotomous key, and we're not going to ask you to construct a dichotomous key. However, the video will help you understand the general idea behind using a dichotomous key. And then open the file of the dichotomous key in lab work 8, which is at the bottom below the case studies right here. Now we can go ahead and open it. It'll be a little small because it's going to open in Canvas. You can click this link and it'll open separately. So it has the gram stain, cocci and bacilli. And then we're looking at the gram-positive cocci here. If it's catalase negative, and then Streptococcus or Enterococcus. And then we're looking at hemolysis. If it's alpha hemolysis, it's either Streptococcus pneumonia or the Viridian Streptococcus. And you can use the Optogen test to distinguish Streptococcus pneumonia from Viridian Streptococcus. Okay, so use this test to identify your case studies. And there's another page here, gram-positive bacilli. Right there. Let's see. So read through that. Those are the different possibilities. And then take a look at case study 1 and use the dichotomous key to identify the species of gram-positive bacteria that's causing the infection in case study 1. Let's go ahead and open case study 1. So we have the patient who came down with a disease. It's actually a 13-year-old teenager who went to a camp, and at home his mother noticed a pustule beginning to develop in the spider bite. And it was very red and inflamed and painful. And then when her son got a fever, she decided to take him into the clinic. And the physician ordered a gram stain and ran routine cultures on the findings. So there's the spider bite, gram stain shown right here. Let me blow that up a little. I think these are the bacteria. and you can see the shape as well as the gram stain. These orange are his cells, the boy's cells. And then they grew this bacteria, it looks like on a blood agar. How do I scroll that over? I'll just shrink it down. Oh, that's not better. How do I scroll that over? Looks like I'm as small as I can do. Oh, there it is. So they did a blood agar test. And as you can see here, if I could only get that to scroll, oh, here it is. Scroll over a little bit. I can pull that up a little. You can see the blood agar is showing beta hemolytic activity right there. And then they did some other tests, the catalase test. There's the result of the catalase test. And then they did the coagulase test. And there's the results of the coagulase test. And then they grew the bacteria on mannitol salt agar. And here's the mannitol salt agar plate. I don't believe we talked about mannitol salt agar in this lab. And then what bacteria was isolated? In a previous lab, I think we talked about mannitol salt agar. So you'll need to go and look what mannitol salt agar does. It, first of all, only grows organisms that grow in a high salt concentration. And then it looks for the fermentation of mannitol. And this is the bacteria here. There's no bacteria there. And so you have to determine if the mannitol was fermented or not. And then identify the bacteria. All right, any questions about any of that? So do the similar thing for each of the case studies. There's 8 of them. So this will take you time to identify each species and then write up on the worksheet what species is causing case study number 1, what species is causing case study number 2, what species is causing each of the case studies. And then when you're done, submit your worksheet. I will warn you that the case studies will be double the points of all the other questions. All right, any questions? If not, I'm going to stop recording and tell you to go ahead and start working on the lab.